Hi everyone, my name is Rhema Uche-Dike. I'm a second year PhD student here at the American Museum of Natural History in Jessica Ware's lab. I'm originally from Nigeria, where I developed my interest in studying insects and particularly dragonflies. So part of my research here at the museum involves using anchored hybrid enrichment, which is a next-gen sequencing method, together with morphological data to reconstruct the phylogeny of a family of dragonflies called Macromidae. I'm also interested in subpopulation genetics in this group, trying to find evidence of gene flow between allopatric populations in this group. Lastly, I'm also interested in using environmental DNA, which is eDNA, for some biodiversity assessments, and particularly seeing how this technique holds up in tropical regions in the study of aquatic invertebrates, because the bulk of the work done on eDNA has been used here in temperate regions and in the study of aquatic vertebrates, and seeing how there's a huge difference in climatic conditions between temperate and tropical regions. I'm looking to see how these environmental factors in tropical regions affect the eDNA yield, and by extension the number of loci we're able to recover after sequencing. And so for me, museum specimens are of particular interest to me and to my research because in evolutionary biology we're trying to see how organisms are different from what their ancestors used to be or used to look like. So having access to a collection here at the museum and in other partner institutions with collections of specimens which are hundreds of years old provides us with really cool insights into these organisms, helping us to answer some really cool evolutionary questions. And for me particularly, trying to study the population genetics, looking at organisms or looking at insects, dragonflies from different regions and in different years, having the luxury of being here at the American Museum of Natural History, which has a really robust collection, providing us with specimens from hundreds of years old and from different regions, allows me to answer these really cool questions that I'm interested in. And on the part of eDNA, so we to validate our results, we need to compare it to results from traditional sampling. So being here at the museum, having these collections which give us results from traditional sampling provides us this essentially provides us with a way to validate our results or my results. So having these collections provide really good insights for us, and it's really valuable for us, especially for me here at the museum.